Thank you all the members of the organizing committee for inviting me to present some of my research work. I am going to present the topic entitled Synthesis, Characterization and Antimicrobial Investigation on some new metal complexes, often aryl substituted aryl chelator. So before starting to my main topic, I would like to spend 1 or 2 minutes for my introduction. You know that I am Bangladeshi. My district is Chittagong over here. So this is the Chinese traveler poet said that a sleeping beauty emerging from mists and water, and given the title a port so grand. This is my city. This is the second largest city in Bangladesh, and you know that the world's largest sea beach in the world, Cox's Bazar, that is situated in my district. And this is the university gate, my Chittagong University. I teach here. And this is the momentum of International Mother Language Day. You know that we fought for our language, Bangla. And this is our Nobel laureate, Dr. Muhammad Yunus of Chittagong University. He was the professor of our economics department. And this is our central library. And this is the building where I carry out my research. Now I am coming to introduce my field. I generally work in the macrocyclic chemistry. And you know that macro means the big, and cyclic, you know that is ring structures. These are the macrocyclic ligand. The cyclic ligand, which contains at least 12 atoms, including 3 or more atoms in its cyclic framework. That means it should contain at least ring of at least 12 atoms, and there should have been at least 3 or more than that donor atoms. So these are the ligands which we deal with. And background of the macrocyclic chemistry, that this scientist Beer first synthesized this one, and this scientist this one. And actually, the sustained development of synthetic macrocyclic complexes starts with the successful preparation of this one. This is the synthetic macrocyclic ligand first to prepare. Let's come to the importance of this field. Why do we choose this field? What is the importance of this field? You know that in the biological sector, you might be knowing about the vitamin B12, hemoglobin, chlorophyll. These are the all macrocyclic complexes which play a multifarious role in our biological system. In medicinal sector, you might be hearing about the valinomycin. These macrocyclic complexes or compounds, they can be used as antibacterial agent, antifungal, anti-cancer, MRI agent. In the analytical chemistry, this cyclic macrocyclic compound, that means 18 crown 6, they can be used as host for the separation of potassium or some other chemicals or some other metal atom, so that these chelators, that means these ligands, can be used for the decontamination of nuclear waste also. We do have a project with the Japan for that. And in the analytical, in other sectors, they can be used in the drugs also. Now, these are the common synthetic routes, mainly 2 routes, template methods and non-template methods. And the template methods means there should be a metal ion should be involved at the beginning of the preparation of the macrocyclic complex. So these methods involve the direct synthesis of macrocyclic compounds from the linear precursors in presence of metal ions. Actually, why do we have chosen this? So some natural macrocyclic compounds, they play a great role in our biological system, like vitamin B12, hemoglobin, chlorophyll. So we think that if we could prepare some of them synthetically, they may mimic these properties or they may have the better properties, because for understanding the metal ion chemistry in these complex structures, it's a bit difficult. So we try to prepare some small macrocyclic complexes or molecules to understand the metal chemistry and other chemistry also. So this is one route, and another route is that first we prepare the ligand where there is no metal ion from the linear precursor. Generally, this route involves the preparation of macrocyclic ligands directly by the condensation of this polyamine, that is, this is diamine, and this is a carbonyl compound. So, by condensation, we can prepare the macrocyclic ligands. Generally, this is another one, another route, and sometimes it so happens that though metal ions can have the, these are the donor atoms, though metal ions can have the coordination number more than the 4, sometimes 6. So we think that if we can put some pendant arm over here, then that can also coordinate with the central metal ion. So thus, these are the N-pendant preparation of polyaza-macrocycles bearing N-pendant groups. So in our research, first we have prepared some of the macrocyclic ligands with just methyl derivative. Then we prepared some N-pendant derivative, and we carried out our research till a long time. So suppose this is the N-pendant group, and this OH group has ability to coordinate with the central metal ion. If there is a incorporation of metal ion, then the metal ion, these groups can satisfy the coordination number of the other than these 4 nitrogen. In this way, some N-pendant derivatives can be prepared by alkylating agents, some of the alkaline disease, ethylene oxide, and some others. Now, what are the objectives of this research? Since I was telling that till starting with this work, we have prepared some ligands which had only the methyl substitution, and later we started to attach some pendant group over here, but we couldn't prepare any aryl substituted ligands. So we tried, and in this study you could prepare this one. This is the aryl substituted group, you can see. And so synthesis of a new methyl and ethyl phenyl substituted trans macrocyclic ligand. Trans because here this double bond is trans to each other. There is a possibility of formation of cis also. And isolation of free ligand. See, this ligand is actually dihydrous, make the free ligand, leaving this dihydroperchlorate group. So, and synthesis of copper to nickel to cobalt complexes from this one, and characterization of the ligand and the complexes by physical, spectroscopic, and other analytical techniques, and antimicrobial studies of the ligands and the complexes. The general characterization of new compounds which we use in our study. First, whatever we prepare, first we test the solubility, then melting point, then elemental analysis, then IR group to detect the functional group, then mass spectra, then proton NMR spectra and carbon-13 NMR spectra, as well as UV visible for ligands and complexes. And in case of complexes, extra most of the time, almost all time, we use the molar conductance and magnetic susceptibility also. So in our just previous studies, we have prepared some of the N-pendant compound, and first we prepared this one, and by alkylating agent it can satisfy the coordination number. So we just confirmed that structure with the X-ray crystallography, which is the best analytical tool. And this is also one more. Sometimes some of the ligands does not allow to put the N-pendant group over here. So this is a case where we couldn't, but for other cases we could prepare some other cases. So these are the you can see this compound, this N-pendant group, it has coordinated with the central metal atom. So it has satisfied the 6 coordination number with 4 donor atoms in the group, in the macrocyclic ring, and 2 from the side chain. So these are the, we have prepared some N-pendant groups and we characterize them. These are the excess structure. And then what we are telling, since we have prepared some alkyl substituted group, that means here only methyl groups. This one also methyl group. In that way, we could prepare, but no one could prepare aryl substituted macrocyclic ligands like that before us. So we tried, keep on trying to do that. So we started to interact with the acetophenone ligand, but unfortunately couldn't, and we have prepared this one, and extractor of this one has been already published. And later we also tried with this carbonyl group and this polyamine, which is the general method for preparing the macrocyclic ligands. That also we thought that we will be able to prepare this ligand, but we couldn't do that, and we got this linear ligand. And later we have used the instead of acetophenone, we started to use carbonyl compounds like benzyl acetone. That means benzyl, this is the ethyl, it is more than that, one more, it is ethyl phenyl group, that whatever it is. So when we found that there is a alpha carbon, if there is no alpha carbon, see, if there is no alpha carbon in the keto group, we couldn't prepare any aryl substituted macrocyclic ligand. So this is our new achievement. We have prepared 2 series. This is the second one. And in our study, we have seen that there is no cis compound form, but in some other cases reported by other researchers, they found that cis compounds, cis means the dime is in the same. We found, you can see that we found the just only trans configuration. So this ligand has been characterized by the IR. You can see that for this ligand, this is the perchlorate band, and other NH band and all groups carbon-carbon all are available in this. And we have characterized by the proton NMR also, and that by the proton NMR we could also characterize the equatorial and axial conformation, and by carbon 13 NMR we could also confirm the number of carbons available in the equivalent carbons available in this ligand. And these are the data, all functional groups. See, band, we got bands for all functional groups, and we could assign the stereochemistry by the proton NMR. We have also confirmed the carbon existed in these ligands. And sorry. Now, general preparation of procedure for metal complexes, general preparation. First, we dissolve the concerned ligand and then metal salt in a suitable solvent, and then we sometimes reflux, and sometimes heating also it gives the complexes. And in some cases, when it's not possible, then we have to use the inert atmosphere. So in our study, we have used the copper, nickel, and cobalt complexes. You know that the cobalt is a central metal ion in the vitamin B12, and nickel in some catalyst. So when we use the copper perchlorate, we got this octahedral structure. When we use the copper nitrate, we got the same octahedral structure, but there is a difference in the conformation. So we got the 2 diastereomers, and that has been confirmed by the X-ray crystallography. And by this way, we could prepare 4 copper complexes, and these 2 are the addition reaction, substitution reaction. That means when we allowed to react this diperchlorate complex with the potassium thiocyanate, then we got this complex by replacing these 2 perchlorates. And in that way, when we use the potassium nitrate, sodium nitrite, then we got this one. Then preparation of nickel complexes, when we interact with the ligand, we carried out the reaction with metal ion, I mean nickel acetate with the ligand, followed by the sodium perchlorate, then we got the square planar structure that we confirmed by different analytical techniques. And this, and when we use the nickel chloride, then also we got the the square planar. It was actually chloride. We tried to replace this one by sodium perchlorate, so we put that also. We could got that also. And then after additions, axial addition reaction, actually when we treated this complex with the potassium thiocyanate, then two axial ligand came over here. So then the nickel
Dr. Tapashi, your device has been muted. Can you please unmute that?
can you hear me, please?
Yes, yes, we can hear you.
Okay. Okay. Sorry, this is the technical problem. Okay.
Okay, not a
So,
problem.
but in the same way, when we treated this complex with the potassium sodium nitrate, we got this one. When we treated this one with the potassium thiocyanate, we got this one. That means the axial ligands are here. But when we treated with the potassium iodide, we haven't got the square planar. That this was the ion exchange reaction takes place. Yeah, anion exchange reaction. Okay. In the same way, when we treated with the cobalt 2 salt, we aerated with the cobalt 2 salt, cobalt acetate with the ligand, and then we added the hydrochloric acid followed by the sodium, potassium perchloric acid. Then we got the 6 coordinated cobalt complex, dichlorido-cobalt complex, and when we treated this complex with the sodium and potassium bromide and potassium iodide, we got the axial substitution reaction products. That means the chlorides are replaced by the nitride group and bromide group and iodide group. So these are the all IR spectral data for all the complexes. You can see this is for the copper complexes. We got the all expected band for the expected groups, functional groups, and these are the data for the nickel complexes. These are the data for the cobalt complexes, and then we have characterized them by the this is the molar conductivity. This is one interesting thing is that, see, this is the octahedral complex and it is the violet one anyway. It has got given the zero conductance because it is non-electrolyte. But when we change the solvent, then color also changes sometimes and it gives the some conductance result. That happens in our study and we have published so many. Sometimes it so happens that for the change of the solvent in different solvents, it changes its geometry. Sometimes from the square planar to octahedral, sometimes square pyramidal. In that way, they changes their geometry, and sometimes that we have confirmed by the color change as well as the conductivity value. And sometimes from that solvent, when we prepare the crystal, we got the different one. From one solvent, we got one geometry, and from another solvent, we got the different geometry. So these are the same thing happened here, and you can see that cobalt. This is the cobalt. It is octahedral. Cobalt cannot have any more than 6 coordination. But if you look at this corresponding to, non-electrolyte. Non-electrolyte means the anion are supposed to go inside the coordination sphere. So that is, we explained for a long time, we couldn't publish our papers also. Then we started to think, then we found that actually the anion coordination, this is the anion association happens though cobalt does not go for the 7 coordination number, but sometimes this perchlorate goes interaction with the chloride in the coordination cavity, then it gives the conductance value corresponding to non-electrolyte. These are all electronic spectral data which matches with our all literature value for all the complexes carried out that. And now let us go for the antimicrobial activities. So this is the, we carried out the antibacterial activities as well as the antifungal activities, and we have our, we don't have the facility for conducting the anti-cancer activities. However, we are working in collaboration with our nearby university. They brought that. So I hope for the next time we'll be able to present the anti-cancer activities results also. Anyway, so these are the bacteria. When alive, it looks like, when it is dead bacteria, it looks like that. So we carried out the antifungal activity against these 5 fungi, and this is the standard antibiotic, the standard antifungal agent, so which is used for the treatment of superficial dermatomycosis disease. And this result actually, when we carried out the antifungal activities for the other ligands and complexes which were alkyl substituted ligands, generally they used to give less value, but in our this aryl substituted ligands, we got the better antibacterial and antifungal activities. Generally, in some of our cases, we got only sometimes 0 also. But here you can see some of the ligands are giving the stimulation also. They are helping for the growth of the fungi. So this is the graphical representation of the these antifungal activities. Let's now come for the antibacterial activities. You see, we have carried out the antibacterial activities of the ligands against 4 gram negative, 4 gram positive